This is Biology for SHS 1, Lesson 1, and this is presented by Ahmed Suleiman Anderson Jr., MPhil Biology student at the University of Education, Winneba. The topic for discussion today is microscope, and then the subtopic is the types of microscopes that we have. At the end of the lesson, students should be able to define what microscope is, should be able to state the types of microscope and give examples of the different types of microscope that we have. So we should be able to define what a microscope is, state the two types of microscope, and then give examples of the type of microscopes that we have. So this is going to be the objectives for the lesson. We are going to be guided by these objectives so that at the end of the lesson we will know whether we'll be able to define what microscope is and state the different types of microscopes that we have. What is a microscope? A microscope is an instrument that is used to view objects that are too small to be seen with the use of the naked eye. So it therefore means that the eye has its own limitation. There are certain things that are beyond the scope of the eye. If something is too small, the eye wouldn't be able to see it, and therefore it is a microscope that is used to view things that cannot be seen with the eye. So we define microscope as an instrument which is used to view objects that are too small to be seen with the use of the eye. So the term microscope can be split into two. We can have micro and scope. The micro in there means small or tiny. When we say something is micro, it means the thing is very small. And then scope is the view. So if you combine the two words together, we say that it is an instrument that is used to view small, that is micro, objects, or small or tiny objects that cannot be seen with the use of the eye, or that cannot be seen with the use of the naked eye. So we understand that generally it is an instrument that is used to view tiny elements or tiny objects. Now let's look at the picture of a microscope. This is a microscope. So this is an instrument that is used to view tiny objects. We are going to discuss its parts later on, but as it is, it is what is used to view objects that cannot be seen with the use of the naked eye. There are so many things about it that helps an individual to view objects beyond the scope of the eye. Now there are tiny objects that can be viewed with the use of the microscope. An example are the different cells that we have. We have the protists, unicellular protists, we have the prokaryotes, we have the cells in the eukaryotes. So generally, the tiny objects that we view in the lab includes the bacteria. Bacteria is a cell on its own, but it's a prokaryote. It's a prokaryotic cell. Then the viruses. Viruses are eukaryotic cells, they lack nucleus. Then we have the plasmodium, it's a protist. The plasmodium is also unicellular, and these are all things that cannot be seen with the use of the eye. Then we can also have the blood cell, the sperm cell in humans. These are all things that cannot be seen with the use of the eye. The blood cells, we have different kinds of blood cells: the white blood cell, the platelet, and then the red blood cells. These are all cells that cannot be seen with the use of the eye, and therefore a microscope is needed to be able to view them. Then we have cells also like the epidermal cell, found in plants, and then palisade mesophyll cells and spongy mesophyll cells. So these are all cells, from bacteria, plasmodium, viruses, um, blood cell, sperm cell, epidermal cell, palisade mesophyll. They are tiny little objects that cannot be seen with the use of the eye, and therefore we should be aided by the microscope, or the microscope should be able to use, or we use the microscope in viewing these tiny objects. A picture of bacteria, we have the Salmonella typhi, we have the Helicobacter pylori, we have Streptococcus, we have Staphylococcus, we have Vibrio cholera, and a host of other bacteria which can only be seen with the use of the microscope. And then examples of cells that can be viewed in the human, as I said earlier on: the neuron, the red blood cell, the white blood cell, the sperm cell, the ovum, the bone cells. These are all cells that can be viewed with the use of the microscope. Now, the brief history of the microscope. The microscope was first invented by Zachariah Janssen. However, the name was coined by Giovanni Faber. Giovanni Faber is popularly known as Faber of Bamberg. He coined the name microscope. Then Antony van Leeuwenhoek was the one who developed the microscope that currently we are using to view tiny objects. He first viewed the detailed structure of the sperm cell and then the blood cell, specifically the red blood cell, and therefore he regarded as the father of microscopy. So microscopy in biology has to do with anything, a technical field that uses microscope to view objects. So investigation into microbes with the use of microscope is called microscopy. So microscopy is the technical field of using microscope to view objects and areas of objects that cannot be seen with the use of the naked eye. Now, the type of microscopes that we have. There are two different types of microscopes. We have the optical microscope, and then we have the electron microscope. For the purpose of time, we are going to look at the optical microscope. Then at another time we take a look at the electron microscope. Now, what is an optical microscope? An optical microscope is a type of microscope that uses light. So it is also referred to as the light microscope. It uses light and a system of lenses. So the light is used to pass through the object, and then the lenses increases the size of the object so that the object can be seen with the eye, because light helps us to see, and then the lenses will also magnify it or increase the size of the object. So as the light is helping us to see the object, the system of lenses is also magnifying them for us to see the object clearly. And therefore the optical microscope is a type of microscope that uses light and a system of lenses to generate a magnified images of small objects. Now, what are the types of optical microscopes that we have? We have the simple light microscope and the compound light microscope. Yes, Daddy. Yes. A simple microscope is a type of microscope that uses single lens to magnify the image of the object. So when you say a simple microscope, we mean the microscope that uses single lens. Now, usually these are the features of the image. The image is erect, enlarged, and it is also virtual. This is as a result of the fact that it uses a convex lens. Now, such microscopes, you can have the magnifying glass. It's a simple microscope. Because we said that a microscope is just an instrument used to magnify the size or things that the eyes cannot see. So magnifying glass, since it contains a single lens, we say that it is a simple microscope. Then we can have the loops, then the telescopes. Then you can have a designed instrument like the microscope itself for viewing objects. So as I said, it has a convex lens with a small focal length, and the focal length of the lens is the distance between the image and then the lens itself. That is the focal length. So the smaller the focal length, the larger the image. So this is a simple microscope. You can see the handle, the eyepiece, the draw tube, the clip, the stage, the coarse adjustment screw, the pillar, the mirror, the base. So within this, the eyepiece contains the lens. So here you could see that it is only one lens that is found within the microscope. Hence, this microscope is a simple microscope. These are also varieties of simple microscope. We have the magnifying glass, that is a simple microscope. Then we have this microscope here. This contains single lens. So that is it. Now, as I said, the smaller the focal length, the greater the magnifying power, and then the higher it be able to increase the size of the object. So usually the magnification of a simple a simple microscope is about 10, which means that it multiplies the size of the object 10 times. So if you have an object which is 5 centimeters long, and then you use the simple microscope to view it, it will multiply it 10 times. Hence, the size of the object becomes 50 centimeters. That is what you'll be seeing, but the magnification would have been or we'll multiply it 5 times. Now, the microscope is used to study soil particles, and that kind of study is called pedology. It is also used by dermatologists to find out various skin types or various skin diseases. Then it is also used in microbiology to study samples of algae, fungi, and the rest. It is used by jewelers to get magnified view of the fine part of the jewelry. Now let's recap. We've seen that the microscope is an instrument that can be used to view objects that the eye cannot see, and we also made mention of the fact that the word micro means small and scope means to observe. So we made mention of the fact that there are certain tiny things that can be viewed with the use of the microscope and which cannot be seen with the eye. Made mention of the cells, the red blood cell, the sperm cell, and the rest. Then we came to the types of microscope. We said we have the simple microscope. So we have the optical and then the electron microscope. The optical microscope, we have two types of optical microscope. We have the simple optical microscope and we have the compound microscope. But generally, they all use light. That is what we, or that is what makes them an optical microscope. So we'll end today's lesson. See you. Bye bye.